Here in the second section of your digestive system PowerPoints, we transition into looking more at chemical digestion and absorption, two of the digestive processes that you have to know in terms of which organs carries out each type. Now we're going to look at the specific details of each one, and we're going to be studying enzymes and macromolecules as we go through. So I think it's important here to start off with some background information. Right? So in this first video here, this is just an introduction to chemical digestion. So to begin with here, I don't know why this is not going away. There we go. We're going to review these digestive processes just briefly. Again, what we're paying most attention to here is chemical digestion and absorption of nutrients. And so you'll see here that we're not going to review each individual digestive organ, but just those that are associated with each of these processes. You may want to take a minute and pause and review which functional classifications or digestive processes go with each organ. So to begin with here, there's four major macromolecules that we study in terms of nutrients for the body. The first are carbohydrates. And so carbohydrates themselves are going to be sugar-based molecules, and these are really important for generating ATP for energy in our body. Now, a few different types of carbohydrates that you may look at. Cellulose, for example, is going to be forming the walls of plants, and this is not something we can digest, so you won't see it listed here on this chart as we talk through it. However, starch, like you would see coming from plants, as well as glycogen, like you might see coming from animals, those can be digested, and so they fit into this category. And so starch from plants and then disaccharides broken down from other sources like fruit and other animals, those can be broken down into short chains of what we would, short chains of carbohydrates known as oligosaccharides and disaccharides. Now a disaccharide is just going to be two of these tiny little molecules known as monosaccharides joined together. And of course, each one of those is named lactose, maltose, and sucrose. So those are those disaccharides. So you take a long molecule like a starch and you break it down into something smaller here. Now that's not good enough for absorption, which is the end goal of this process. And so you'll find that each one of those disaccharides has to be broken down into something even smaller. Now these smaller components are known as monosaccharides, and they are galactose, glucose, and fructose. Now, if you take a look at the arrows here, you can see most of them point to glucose. Glucose is the sugar or the monosaccharide of choice for most of the metabolic processes in your body. And so there's two reasons I have you look at this slide. The first is just to understand how you start with a larger macromolecule and you break it down into smaller components, becoming familiar with the terminology of each stage as you go. The second reason I show you this is because you can see the different stages at which chemical digestion of carbohydrates can take place. It begins in the mouth and in the small intestine using two different types of amylase. And that's what's responsible for breaking it down into these first chunks of oligosaccharides and disaccharides. But you don't actually see the hydrolysis take place in forming smaller disaccharides and monosaccharides until you reach the small intestine's brush border enzymes. And so those enzymes we're going to find finish the job, and they actually give us these tiny little monosaccharides here. At the very end of digestion, of any chemical digestion, the goal is to have molecules that are small enough that they can be absorbed across the wall of the small intestine. And so this is the process for carbohydrates. Now, it's a similar process for proteins. Proteins themselves are very large molecules here, and you want to break those down into large polypeptides. A polypeptide itself is basically just a long string of amino acids held together by peptide bonds. And the difference between a polypeptide and a protein is one of these long strands coiled up into a ball in many cases. So this protein, it needs to unravel and be broken down to a polypeptide. And large polypeptides, long chains, are then cleaved into smaller polypeptides or even small peptides, very tiny fragments of 2 or 3 of these amino acids all together. And finally, at the very end of chemical digestion, you want these divided into amino acids for the most part. And amino acids are the individual building blocks for a protein, tiny monomers, if you will, that can be absorbed very easily across the small intestine. So like we saw with carbohydrates, you can see this process actually begins here in the stomach. So the stomach begins by using pepsin in the presence of hydrochloric acid to unravel and begin to break down these proteins. In the small intestine, you start to see more enzymes like trypsin, chymotrypsin, carboxypeptidase break down large polypeptides into small polypeptides. But then once again, you finish the job using brush border enzymes of the small intestine. These are going to be named aminopeptidase, carboxypeptidase, and dipeptidase. When you look at lipids or fats, you find that like your carbohydrates at the very beginning, this kind of digestion can start way up in the mouth. So you begin here with unemulsified triglycerides. Now, unemulsified itself indicates that they are present, but they are not mixing well with watery secretions. As you all know, oils and water don't mix. And this is a really big problem because all your digestive enzymes, like the lipases that you see here, are all water-based. And so it's almost like you're taking a water hose and you're spraying it at a fat globule. You're probably going to make contact with the triglycerides on the outside of that fat globule, but you're not going to do anything to the internal components of it. So you're just not going to digest it or break it down well. And so you can see all along here in the mouth and in the stomach, these are unemulsified triglycerides, and you're doing your best throwing some lingual lipase and gastric lipase at them to try to break them down. Where the tide turns is when you reach the small intestine. And this is where you see emulsification, right? And what this means is that these triglycerides are finally becoming mixed with these watery enzymes that can break them down. Now, this is carried out by bile. Right? The bile salts in bile are not enzymes. They are not responsible for digestion, but they are responsible for emulsification, mixing all these lipids or triglycerides with watery enzymes. And so at the very end of the day, you finally emulsify these triglycerides and in one last-ditch effort, throw some more enzymes at them. These are pancreatic lipases in the small intestine, and that does the job. You break down these triglycerides into monoglycerides. This is the backbone of a triglyceride, and tiny fatty acids. And so these, again, are the smallest components that are going to be absorbed at the very end of chemical digestion. And finally, you have nucleic acids. Now, if you're not familiar with it, nucleic acids are DNA and RNA, and all cells contain, or almost all cells contain nucleic acids. So no matter what you're eating, you're probably bringing a nucleic acid. And these are already quite easy to digest, and so you don't need quite as many steps, but they will be broken down into pentose sugars, nitrogen-containing bases, and phosphate groups. And that's going to be carried out here in the small intestine using larger enzymes here, like ribonuclease and deoxyribonuclease, and then brush border enzymes like your nucleosidases and phosphatases. Once again, this is what can be absorbed at the very end of chemical digestion. And so as a review, during chemical digestion, starch is digested into, choose the best answer. Right? So is it digested into monosaccharides, such as glucose, amino acids, triglycerides, or pentose sugars with nitrogen-containing bases? Question 2. Amino acids are small monomers of which larger macromolecules? Is this of carbohydrates, proteins, lipids, or nucleic acids? Question 3. Which of the following macromolecules will not be chemically digested by secretions in the mouth? Carbohydrates, proteins, lipids, A and C only, right? Or E, all of the above. Question 4, which of the following digestive processes is facilitated by the large intestine? A, propulsion, B, absorption, C, chemical digestion, or D, only A and B, or E, all of the above. In the second video, we're going to look at chemical digestion, beginning with the mouth and moving to the stomach. You won't want to miss it, so I'll see you there.